Hello, I'm Nick Carpita, formerly of Purdue University and now at the National Renewable Energy Lab in Golden, Colorado. Titled my talk is Redesigning the Plant Cell Wall for Fuels, Chemicals, and Materials. This is a long-term effort that was started by our Energy Frontier Research Center called C3 Bio. And we had the theme always of no carbon left behind. We looked at biomass as a total set of materials, of molecules and materials that are useful, and we set out to rescue all of them. Biomass recalcitrance, we have assumed to be an atomic scale property, and it's property of lignin occluding cellulose from digestion by cellulases. But recalcitrance, in the sense of deconstructing biomass, has many more properties than that, because when we look at the cell wall components of cell walls that we are interested in as a source of fuel intermediates for liquid hydrocarbons, these are constructed in a nanostructure cell wall that has several orders of magnitude of tissue organization. And it'll turn out, as I'll relate to you, that the middle lamella, the way cells are cemented together, are just as much a part of recalcitrance than lignin-occluding cellulose. We wanted to first say about the recalcitrance of cellulose, and that is asking how big are cellulose microfibrils. And we had worked out or used a tempo-catalyzed oxidation, which is well known, that oxidizes any solvent-exposed glucose 6-hydroxyls to hypochlorite oxidation in the presence of a tempo catalyst to their respective uronic acids. These can be activated by a diamide and reduced with borodeuteride. And those that are oxidized can then be revealed by electrospray ionization, mass spec. The anhydrous within the crystal are not solvent exposed, so they never get deuterated. Amorphous, all of the cellulose, all of the glucoses are reduced. But in surface chains, only every other one is reduced, and therefore, where you have one that is oxidized, the next one is that group is tucked back into the crystal and avoids oxidation. And so, when these surface chains are produced, we can see them. Let's see if I can get this out of the way. Okay. That the surface chains have either an aldehyde or one of the glucoses is reduced when you solubilize the cellulose and digest it with cellobiohydrolase to cellobiose units. Amorphous is very small, and when you correct for C13 spillover, the surface chains are not that much. In fact, most of the material is anhydrous. If we work with the current models of cellulose microfibrils that are predicted to be 18 or 24 chains, some as high still hold that they're as high as 36 chains. The ratio of anhydrous to surface is really small. Only in 36 chains do you have more anhydrous chains than surface chains. But our data is saying, regardless whether you have secondary walls of wood or cotton, or the primary walls of a Arabidopsis leaf or maize coleoptile, that the anhydrous to surface ratios are much larger than one would have thought with microfibrils of only 18 to 24. In fact, you have to put 6 to 8 of them together to get to the ratios, some of the ratios that we're seeing, depending on the type of microfibril that is synthesized. And therefore, this adds greatly to the recalcitrance of these bundles. One of the ways that we were able to break up these bundles and dissolve cellulose is with neat trifluoroacetic acid, where we soaked cotton linter cellulose overnight at -20 degrees in 99% TFA that causes the cellulose to swell greatly. And if you then heat it, you get a pourable liquid that can crash and form gels. These gels are easily digested by the C-TEC2 cellulase preparation or cocktail that we're using. And heating, that really doesn't add that much to it. Now, if we look and bring in lignin, for several decades now, scientists, particularly Clint Chappell and Sean Mansfield, UBC and Purdue, have been showing that if you modify ferrylate hydroxylase to knock it out, then you get a poplar or Arabidopsis that is close to 100% G-lignin. If you overexpress it, they were surprised to find that 90% of the lignin becomes in the S-lignin, which can only form beta O4 linkages, makes it very easy to digest out of the wall, and it doesn't occlude cellulose as well, is the interpretation. A mole-methyltransferase is also necessary, and if you block that but overexpress F5H, you then get the hydroxyl form, or the 5-hydroxy G-lignin. Regardless, what many people have found is that unless you pre-treat by steam explosion or steam expansion or AFEX or in this case by maleic acid hydrolysis of xylans, then you only see the effect of the high S after these kinds of pretreatment. So there's something else going on besides the limited occlusion of cellulose. The other way to look at lignin, of course, is to depolymerize it gently using a zinc palladium or a nickel catalyst. And the advantage to that is that you actually preserve the aromatic rings and you can recover these as a product. And that's kind of important in terms of the lignin first biorefinery, is that you're actually not treating lignin as a problem, but as a product. And this, of course, this leaves cellulose pretty much intact, and as well as the non-cellulosic polysaccharides. Well, if we look at lignified materials and we try to use the TFA-induced swelling, we found that the lignin in this case really blocked the swelling of cellulose. You had to heat it in order to break up that lignin cellulose interaction. However, in the catalytically delignified materials, you can see they swell quite readily by simply putting them in TFA at minus 20 degrees. When we crash these into ethanol and recover the materials, and we look at how they digest, it's the same thing with untreated materials. They don't swell in TFA, and therefore you don't get an improvement in hydrolysis. You also don't see an effect of whether you have high S or high G or high hydroxy G lignin. However, in the delignified materials, you then see a huge increase in the digestion with this C-TEC2 cocktail. And there you see a lignin effect. Even though there's no lignin in the material, the S-lignin is easier to digest. Even though when we look at the amounts of anhydrous cellulose chains, they actually increased after CDL treatment. So that cannot be related. These differences cannot be related to occlusion of lignin in cellulose, and we look elsewhere. And the elsewhere turned out to be pectin in the compound middle lamella. In wood, it is the middle lamella is a amalgam of pectin, of lignin, and some xylan material, not cellulose. And so we looked at this crudely by taking wood particles of poplar And these particles, if we could we treated with acid chloride to oxidize the lignin, and then followed it with a pectin extraction with dilute alkali, and we could get single cells. It didn't matter which we did first. If we took the CDL material that had the lignin removed from it, and then looked at the impact of xylanase, we see that it is starting to fragment these particles, but you simply don't have single cells. You only get single cells if you follow that xylanase treatment with RG lyase, which will digest this polymer. This pectic polymer, RG lyase, is part of the tensile strength and a major part of the pectin material. We overexpressed an RG1 lyase, particularly one that's expressed in these transgenic poplars. As you do with transgenic plants, you get various levels of expression of this. And in partially oxidized lignin and partially extracted pectin, the RG lyase expressing lines gave a much greater cell separation. And therefore, this kind of transgenic plant is very much related to the comminution that is required in order to break down biomass, and undoubtedly contributes to the inability of S-lignin to make a strong compound middle lamella, and increases cellulase digestion, not by removing the occlusion from cellulose, but allowing greater access through the cells. And finally, just what does all of this mean for the biomass and the new bioeconomy? We look at the materials that are found in the cell wall, and we are looking at ways to gently remove them, either intact. All of the effort up until recently has been generating molecules. But what we really want from biomass is not only molecules, but materials. As we look to replace everything that we now make with petroleum with a bio-based materials, we look to nanocrystals or nanofibers as substrates to which we can attach functional groups. One of the ways of creating functional groups is our functionalized hemicelluloses, where you can put fluorescent tags. You can oxidize galactose residues on galactomannans or the galactose residues in xyloglucan and attach and use them as functional groups to attach residues. And then you can put them on cellulose nanocrystals or nanofibers to create all kinds of materials that are used not just as fiber, but as components of electronics and so forth. And with that, I would like to thank you for your attention. I want to call out, there's a lot of people, both at NREL and at Purdue mostly, that worked in this project. I want to give a little shout out to Tanya Shiga at University of Sao Paulo, funded by CAPIS on a project where she looked at the TFA solubilization, how to break up the crystallinity of cellulose. Our center leader is Maureen McCann, and I followed her out to NREL, and she's now director of the Biosciences Center, and we're looking at ways of incorporating synthetic biology into this new age of biomass and the bioeconomy. And notice our email addresses have changed, and be happy to answer any questions that you might have. Thank you.
Bom dia. My name is Lee Lynd. It's a pleasure to be with you today. I am talking to you from my home in the northeastern United States, where the snow recently melted, and I look forward to the pandemic getting under control so that I can visit my friends in Brazil. And I thank the meeting organizers and also the session chairman, Marco, for the invitation to speak, which I'm grateful for. So, moving on in a 15-minute presentation, I'm going to talk to you about consolidated bioprocessing with co-treatment, abbreviated C-CBP. an alternative cellulosic biomass deconstruction paradigm. So for the purpose of this presentation, I'm going to assume that second generation biofuels are a good idea. The arguments are familiar to this audience. We also need to acknowledge challenges and the changing biofuel landscape. Deployment has fallen far short of expectations over the last decade and led to radically decreased investment. And most of the world is not looking to biofuels for carbon neutral light duty transport today. For the last half century, the vast majority of effort on biological deconstruction of cellulosic biomass has focused on a scientific and technological paradigm featuring thermochemical pretreatment and added enzymes produced by aerobic fungi, in the hoped for event that this paradigm proves sufficient to launch a robust second generation biofuel industry. There's still rationale for considering alternative paradigms so that we can lower cost, increase competitiveness, and accelerate deployment. But in the event that the current paradigm is not sufficient to launch a robust biofuel industry, which I think objectively hasn't happened yet, then the alternative paradigms are the only way forward for 2G biofuels. We should learn from other industries. If you look at batteries, they've changed chemistries multiple times and targeted a staircase of applications. If you think about film versus digital cameras, sticking with the old paradigm did not work out too well. Well, let's think about how we got to where we are, and many people are familiar with this story. At the end of World War II, because canvas tents weren't lasting very long in Southeast Asia, the US Army Natick Labs initiated research into microbial cellulose utilization. But when synthetic fibers emerged in the mid-60s, that refocused work on food and fuel production. And early on, they asked what microbes are the best cellulase secretors. Their answer was aerobic fungi. This was pre-genetic engineering. So you basically had to look for what nature already produced. It was also asked early on, what can be done before biological processing to enhance enzymatic solubilization? Milling prior to fermentation and hydrolysis was deemed to be too energy intensive, and thermochemical pretreatment was found to be more promising. This led in about 1980 to the current paradigm being established, and processes that look like this with fungal cellulase production and thermochemical pretreatment. A lot has happened since then, as you know, including in the last 5 years, about a half a dozen pioneer cellulosic ethanol plants coming online around the world. Most, if not all, have yet to operate at full capacity. None have been replicated to my knowledge, and most have been shut down or repurposed, unfortunately. So I'm going to talk about a different paradigm, consolidated bioprocessing with co-treatment. And I point out in particular that it avoids the two factors that are responsible for the high cost of the conventional paradigm, namely thermochemical pretreatment and added enzymes. But I also acknowledge that this alternative paradigm entails new challenges. So when I speak of consolidated bioprocessing, this concept eliminates the need for added enzymes by using anaerobic microbes that produce saccharolytic enzymes while fermenting carbohydrates to ethanol. We have 4 biologically mediated events: enzyme production, deconstruction, and fermentation of C6 and C5 sugars. CBP aims to do those in one integrated unit operation. Well, it's pretty logical to ask, what is nature's best when it comes to deconstruction of cellulosic biomass? There are lots of systems and lots of architectures and lots of really interesting science. These dots here are yeast. They are not enzymes, but it demonstrates that if you do simultaneous saccharification and fermentation with added enzymes, the enzymes, which you don't see here, act on the substrate surface, and the product, the solubilization products, the sugars, diffuse away to the microorganisms. That's not the way, however, nature does business with cellulolytic microbes. They actually stick to the fiber, as shown here for Clostridium thermocellum. We could have asked a long time ago, but didn't, which of these systems is most effective? And there's some reasons that that was not asked. It's not a simple point estimate or point comparison. It's nuanced. It's never been easy to get funding for. And there's the risk your conclusions might make more enemies than friends. And in science, we tend to respect other people's model systems. But we jumped in about 5 years ago And this was some of our very early data, a paper by Julie Pei. And we were somewhat surprised to see that a fungal cellulase and yeast system, more or less representing the status quo, performed rather not anywhere near as well as a pure culture of cellulolytic anaerobes, in this case, Clostridium thermocellum. It's also interesting to note that C. thermocellum did as well as a horse manure enrichment, but that's a story I don't have time to go into today. But since then, we've undertaken extensive comparative studies over a broad range of variables. Indeed, to my knowledge, the most extensive comparative studies that have been undertaken to date. And here's just a summary of the kind of thing we see. So here on switchgrass, comparing fungal cellulase with SSF and C. thermocellum, we see that in every case for these different variants of switchgrass, developed to be less recalcitrant, by the way, that the bacterial system does several fold better, up to 6-fold better than the fungal system. Down here, if we look at corn stover, 3-fold better. If we look at corn fiber, twice as good and virtually complete solubilization. Interesting, if we look at Avicel, fungal cellulases can't completely solubilize even a model microcrystalline cellulose, whereas C. thermocellum just blasts through it. So C. thermocellum is 2 to 6-fold more effective than industry standard fungal cellulase at plant cell wall deconstruction, and this is all with no pretreatment other than autoclaving. Well, if we think about why this might be true, and that's still a book that hasn't been completely written, but everyone's trying to figure it out. First of all, recognize that when we study enzyme kinetics or learn about them classically, we learn about enzyme substrate complexes. But in the case of cellulose utilization, it's very clear that it's done in nature by enzyme substrate microbe complexes. And there's strong quantitative evidence that the microbe is not a passive partner in that and actually accelerates the rate. The local physical chemistry where the reaction occurs is impacted by the cell surface, and the whole system has been subjected to selective pressure for surface occupation and product uptake. It's interesting to note that the C. thermocellum cellulase system or cellulosome is gigantic compared to all the catalytic components of the non-complex enzyme systems, such as produced by T-ricei. And what's found is that the ends of fibers, when attacked by these smaller fungal enzymes, it's sort of like sharpening a pencil, but there's no ability to actually part the fibers. By contrast, with C. thermocellum, it's able to splay the ends of these microfiber ends. And in fact, if you make a mutant of C. thermocellum that lacks this CYP-A protein, it's no longer able to do that. But then another question is, so how best can we give biology a hand? And the field's answer for 35 or 40 years has been combination of heat and chemicals. Nature does it differently. My friend Everson from Unicamp told me I should never give this presentation without showing the chewing cow. So there we are from my son's farm. But I point out that rumination, that is the alternate chewing and biological attack, which is exactly what a cow does, increases the surface area by up to 10,000-fold, according to this review by my friend Paul Weimer, and actually uses 2% of the metabolizable energy of the cow. Well, if we're going to rethink our deconstruction and our biocatalysts, we ought to rethink the augmentation as well. So if you look at yeast, yeast is among the most resistant cellular life forms to chemical inhibition. That's why you can inoculate an ethanol plant in Brazil once a year, expose it to sulfuric acid, the cells in the recycle line. And since yeast is tougher than everybody else, you can maintain your functional culture. Remarkable. Cellulolytic anaerobes, such as C. thermocellum, are not particularly resistant to chemicals, but they are among the most resistant cellular life forms to mechanical disruption. So when I talk about co-treatment, I'm talking about mechanical feedstock disruption during fermentation in lieu of thermochemical disruption prior to fermentation. And you can readily show, this is a model system that we use to look at co-treatment. It's not how we envision it would be implemented industrially, but essentially we have a little piece of 4-inch process pipe. We fill it with stainless steel beads and stir it so we can mill the material. We first did tests with soluble substrates and demonstrated that in the presence of milling, fermentation is totally halted. whereas C. thermocellum is not affected very much. That's because it's got a smaller and tougher, thicker cell wall than yeast does. And now if we revisit this earlier plot I showed, first of all, I show the incremental benefit compared to controls of a 10-year effort actually to develop less recalcitrant plant lines, in this case for switchgrass. So relatively modest impact, I would say. But if you now look at the impact of doing the ball milling fermentation, a form of co-treatment, we see that for both woody and herbaceous feedstocks, we're able to get to well into the, generally into 90+% of the carbohydrate solubilized without pretreatment and without added enzymes. And it turns out, as many people are familiar, that the physical properties of lignocellulose change really radically in the early stages of fermentation. So we documented in a few years ago that there's a 2,000-fold decrease in viscosity. This is a log to the base 10 plot here that occurs during fermentation by C-therm. So if you want low milling energy, it's really helpful if the material is partially solubilized. But the fungal cellulase is not very good in the absence of pretreatment. C-thermocellum is much better, so you get this overlapping region. So rather than non-biological disruption before biological processing, pretreatment, we're actually have biological attack and then non-biological disruption. I just want to acknowledge that to make this idea work, we don't have time to go into it, although it's a pleasure to. We need to engineer the thermophilic microorganisms to be better at producing ethanol and behave as industrial catalysts. And there's quite a bit to that. This just shows, if you look at ethanol yield and concentration from where we started, in some cases, we're into the feasible region. You actually can't make much more than about 60 grams per liter ethanol from lignocellulose, even though you can from sugarcane and from corn. And for C. thermocellum, we're getting there, but have a ways to go still. In terms of techno-economic analysis, we did a paper a few years ago with friends and co-authors from NREL, and we looked at various technical scenarios. And if you look at the payback time, essentially the capital divided by the annual operating cost, for the conventional paradigm, even at 100 million gallons a year or about 400 million liters per year, we're looking at 20-year payback, which is a long time. And if you just go right to the advanced CBP case, first of all, it's about an 8-fold shorter payback. And secondly, we have economic feasibility at about 10-fold smaller scale. And in fact, 25% higher greenhouse gas emission reduction per ton. So in terms of take-home messages, there are good reasons to investigate alternative scientific and technological deconstruction paradigms, as I talked about in my introduction. C-CBP using engineered thermophiles is one such paradigm. It avoids the two factors responsible for the high cost of current technology, added enzymes and thermochemical pretreatment. It uses deconstruction biocatalysts that are decisively better than commercial cellulases in controlled experiments. It involves co-treatment, that is milling during fermentation, which enables high fractional carbohydrate solubilization by growing cultures of cellulolytic anaerobes. Techno-economic analysis indicates potential for disruptive cost reductions, and making C-CBP practical requires metabolic engineering of thermophilic anaerobes to improve product formation, further validate co-treatment, integrate process components, and establish robustness. And I'd just like to close, in addition to thanking you for your attention, by acknowledging the organizations that are working on this: my lab at Dartmouth, the Bioenergy Research Center in the US, funded by the US DOE, a startup company, Enchi Corporation, and in particular, a newly formed lab at Unicamp, supported by FAPESP, the Advanced Second Generation Biofuel Laboratory. So thank you very much. It's a pleasure. I'm not quite sure what kinds of discussion we will be able to have with the virtual format, but I look forward to anything we're able to do. And obrigado.
Hello, everyone. I am Dr. John Hannon, COO and founder of Redesigning in Irvine, California, and I'm going to present to you reducing transportation carbon through low-cost consolidated alcohol deoxygenation and oligomerization, the CADO process, to gasoline, diesel, and jet fuel blend stocks, as well as higher value chemicals, BTEX. Okay, when we look at CADO transition from, to alcohols from sustainable sources, on the left you look at different feedstocks that this technology can handle, ranging from sugarcane to corn or starch, wood chips or cellulosic biomass, to carbon dioxide as well. And on the left, greenhouse gas reductions are highlighted in blue, where sugarcane about 65% or higher, corn feedstocks near 50%, cellulosics being 70% or greater, and then flue gas or carbon dioxide being 90 to 100% plus. And through various technologies, current first generation, through fermentation, some emerging second generation technologies, and the future processes, you look at in the yellow, fermentation, thermal conversion, electrochemical conversion of carbon dioxide to make these various alcohols, ethanol, butanol, methanol, or different mixed alcohols. And then those products can be then converted using our CADO process in the blue there to make the variety of different products you see on your right. So anywhere from plastic precursors, that's the BTEX, benzene, toluene, ethylbenzene, xylene, to LPG, liquefied petroleum gases, mainly propane, for example, high octane gasoline, jet fuel, and diesel fuel. Okay, when you look at the energy flow for the transformation from sugar to hydrocarbon fuels, in blue you see the kind of formula that we present. Well, on the left you see the ethanol molecule can be represented by C2H5OH, or you can look at it as C2H4H2O, and that gets converted into hydrocarbons as C2N H4N plus water. And on the left, what we start with is, if you look at a glucose molecule, or 1 kilogram of glucose, 15.6 megajoules of energy, that typically goes through a fermentation process where the glucose is converted into ethanol and carbon dioxide. The carbon dioxide comes out the bottom there, you see, and it really comes out the top, but it has no calorific value. And about half of the weight of the glucose is represented in the carbon dioxide that's released. The remainder, about 0.51 grams of ethanol, has the energy content about 15.2 megajoules. And so the majority of the energy in the glucose is conserved into the ethanol molecule through fermentation. And you see through the fermentation, carbon dioxide does remove a significant portion of the oxygen that's present in the glucose. And then if you take that ethanol and then run that through our catalytic reactor, what our process does is removes the remaining oxygen in the form of water out the bottom. And you can see the water has no calorific content, 0 megajoules, and represents about 2.2 out of that 0.5 kilograms, roughly 40% of the mass. And the remainder, 0.31 kilograms, ends up as fungible fuels and gases, or hydrocarbons, as you see in the equation above. And again, the majority of the energy content in the fungible fuels and gases is conserved from the ethanol molecule. So this technology removes the remaining oxygen and really densifies or increases the energy density of the ethanol into fungible fuels and gases, although the volume is smaller. Some of the technical advantages of the CADO process and also commercial advantages are, reading them off on the left side there, there's 7 bullets. It's a single reaction system, so converting alcohols into hydrocarbon products, single stage reaction system. We always get 100% conversion of the ethanol. Another distinguishing feature is it operates near ambient pressure and moderate temperatures. We have no external hydrogen that's added to the process. It's a low capex and low opex solution, and that's another distinguishing feature that makes this really cost effective. And flexibility to shift products. It gives the ethanol producers the ability to, make ethanol if it makes sense. Otherwise, you can take the ethanol and convert it into different products if it makes financial sense. It offers the ability to debottleneck some biofuel production. I'll get into that a little bit later. If the plant is bottlenecked at the distillation column, this allows it to debottleneck it by removing the ethanol water vapor from the distillation column. These fuels are also fungible. You can use the existing pipelines versus ethanol that you typically have to truck out or rail out. And again, as I covered before, most of the energy in the ethanol is conserved in the hydrocarbon products. Another distinguishing feature is these technologies can be scaled up directly from pilot or demonstration reactors. And I'll get into that a little bit later because the vapor phase heterogeneous catalysis offers really large scale-up factors. When we look at ethanol as a molecule, you know, it's the most produced biofuel in the world. So in the United States on the left there's about 220 plants that produce roughly 15 billion gallons of ethanol. In the middle there in Brazil, produce just under 400 plants producing about 7 billion gallons. And then in the EU and really across the world, there's in the EU about 1.5 billion gallons. So there's a really large platform out there. And it's a very clean, renewable, low-cost fuel. That's the molecule that we're focusing on to launch this technology. Yeah, we work with a company called Technip FMC. We chose to work with them to accelerate the CADO commercialization for a number of reasons. First, they're a very large company. They have a worldwide presence, and they specifically have extensive success on the third bullet there in directly commercializing a wide range of catalytic technologies based on their demonstration reactors. So they've successfully deployed over 20 commercial technologies, catalytic technologies. You see there in the bottom left, Technip demo operations picture of our demonstration reactor. And then they're able to scale up with guarantees 1,000,000x from that demonstration reactor to a commercial plant. And so a combination of their expertise, their commercial successes, and ability to scale up with guarantees really attracted us to work with Technip. Yeah, we entered a program with the Department of Energy and Technip to really advance this technology over a 2-year period. You see a picture on the top right of our pilot reactor system, and the bottom right looks at the kind of the technology advances I'll get into in a second here. But initially, what we did was we successfully transferred the CADO technology from Oak Ridge National Labs, where it was invented, to Technip FMC. So that was our first advance. And again, this is highlighted in our National Academy of Science proceedings of the National Academy of Sciences paper, and references down below. Secondly, the other thing we've done is we advanced the catalyst from powdered form, very small scale, to commercial extrudates while cutting costs. So that was another catalyst advantage. We also increased the liquid hydrocarbon yields, defined as C5 plus hydrocarbon, or basically a liquid at room temperature and pressure, from initial 36% to greater than 80% in roughly an 8-month time period. And it's important to do that from a economic analysis, as you can, the liquid fuels are more valuable than the gaseous fuels. The fourth bullet, we also scaled the technology up 300 times in the 12 months while increasing the yields. We always maintain 100% conversion of ethanol and a single step operation with no external hydrogen. Also maintain the mild conditions, 350 C, and just enough pressure, roughly 60 psi, to push it through the process. We also looked at different wet ethanol feeds, so we can range it from 25% to 100% ethanol in the feed. Some of our products highlighted here, you can see a combination of aromatics, aliphatics, and olefins. On the left scale, you see kind of time of operation. The dotted line shows our regeneration process and the various products and liquid yields, very steady. And on the right, you can see the breakdown of a typical run that we offer for our gasoline product. Also in this national proceedings of the National Academy of Science paper, we looked at the comparison of on the top, a typical ethanol plant that makes 100% ethanol, and on the bottom, if we were to bolt on to this this ethanol facility, what would it look like to take that ethanol at a 40% off the beer column and convert it into fungible fuel blend stocks? So we did that comparison of that paper as well. And we looked at the cost of this conversion over the months of development of this technology. What we found is that we're projecting that the cost on the right, cost of that catalytic conversion is really approaching the same cost of ethanol dehydration. So really, it's very interesting, gives the ethanol producer the ability to do or to produce ethanol or to produce hydrocarbon fuels at the same cost. And so this kind of just highlights it's about $1.46 per gigajoule for the conversion process into 100% ethanol. We're at about $2 per gigajoule now to make fungible fuel stocks, but we're projected to be very close, if not under, the production cost of 100% ethanol. Now, this technology works for a variety of different feedstocks, as I mentioned before. On top left, it works for a corn wet mill. Bottom left, cane sugar as well. And then on the right, cellulosic ethanol, and kind of in the middle right, more of a corn dry mill. And you can see how they kind of integrate in with the different processes. So those 4 different conversion processes and feedstocks. We also looked at how this technology compares to producing hydrocarbons through petroleum. So we looked at different ethanol feedstocks on the left, some cornstarch, cane juice with current and projected technologies, as well as cellulosics with current and projected technologies. And we looked at, what the minimum blend stock selling price for these fuels would need to be from these various feedstocks, and we compare them to the oil at $60 and $100 per barrel. And what we see is that there are instances where the technologies can compete with $60 per barrel oil, namely cornstarch under projected technology. And then later on, we have cellulosics that are able to compete with $100 per barrel oil, and that's without incentives. We also looked at the payback for $60 per barrel oil, $80 per barrel oil, $100 per barrel oil for these different technologies if you were to bolt them on as a function of different incentives. And what we've seen is a wide range of paybacks. So, $60 per barrel oil, it's NNR, means that, really can't pay back, no net return. But as oil price increases and you look at the various incentives, we start to see payback periods for these bolt-on technologies. And then certainly at $100 per barrel, these really start to compete. So that's how the incentives really impact the payback periods. We also do look backs, and we look at if ethanol producers had this technology, and we look back in the past, would applying this technology make sense or not? And we look at the oil price, the ethanol price, and if you were able to convert ethanol into diverted fuels, when would it make sense? And what we find out is that these real large pockets are windows of opportunity where you can really increase the margin by taking ethanol and converting it into these different diverted fuels. But there are instances as well where you just produce the ethanol. We also looked at the greenhouse gas reductions with this technology, and what we found is with these various feedstocks, we found is that first generation feedstocks offer roughly 40 to 69% greenhouse gas reductions, and from the second generation greenhouse gas reductions range from roughly 70% to about 96%. So some of our take-home messages for this technology is this novel CADO technology really converts ethanol and other alcohols, I didn't get into that much, but into gasoline, diesel, and jet fuel blend stocks, as well as these higher value chemicals without added hydrogen. This technology offers a low capital and operating cost based on its simple technology. The technology also offers greenhouse gas reductions ranging from 40 to 70% for first generation and 70 to 96% for second generation. We can produce hydrocarbon fuels at about the same cost per energy as producing anhydrous ethanol. Product can be competitive with petroleum-derived equivalents with various incentives. And the technology will be ready in months, not years. And we're able to also produce alcohols that could be produced from carbon capture to the renewable hydrocarbon products. And thank you very much.
Hello, my name is Boris Stambuck. I'm professor of biochemistry at the Federal University of Santa Catarina. And in my presentation, we will show you the work we are doing on the expression of heterologous xylose transporters in Saccharomyces cerevisiae. As you all know, regardless of which biomass is going to be used by different countries to make biofuels, xylose is the second sugar more abundant in nature. So any process will need to use this sugar to produce biofuels. So there has been a lot of efforts into introducing xylose utilization pathway in cerevisiae, although cerevisiae has his own pathway for xylose, but it's not as efficient for production of ethanol. So in this work, we used integrative plasmid to overexpress the 3 genes that are required to metabolize xylose reductase, xylose dehydrogenase, and xylulose kinase, which convert xylose into xylulose 5-phosphate and then into ethanol. But what is the limiting step? Obviously, the limiting step is transport across the plasma membrane. And as you can see in this figure, in the right side, in the left side, you can see the consumption of glucose, black symbols, and production of ethanol. The consumption of xylose, which is lower and less ethanol. But the main problem occurs when you have a co-fermentation of both glucose and xylose. In the right side, you can see the presence of xylose slows down the consumption of glucose, and also the presence of glucose slows down the consumption of xylose, because glucose and xylose are competing for the same transport system. So in cerevisiae, there are over 20 different sugar transporters, exhausted transporters. But if you delete the 8 major sugar transporters from 1 to 7 and GAU2, you will get an exhausted transporter null strain, which will not consume glucose, fructose, or xylose. And so we used this exhausted transporter null strain, transform it with the integrative plasmid to analyze the influence of individual exhausted transporters in glucose or glucose-xylose co-fermentation. So our results indicated that no cerevisiae exhausted transporter is suitable for xylose fermentation, which means that we need new xylose transporters that obviously that can be isolated from other yeast species which efficiently ferment xylose. So in a first effort, we used this exhausted transporter null strain overexpressing the xylose metabolizing genes to screen a genomic DNA library from Saccharomyces GPT. This allowed us to isolate transformants directly in xylose plates, and that allowed us to isolate 4 different plasmids that could allow the cells to grow on xylose and other sugars. It turned out to be that these 4 plasmids were 3 different genes, and 2 of them were never being described as xylose transporters. But the best transporter was the SHOOT1 gene from SGPTs, which allowed efficient consumption of glucose and xylose, or even during co-fermentation. So, but we were also interested in this, discover new sugar transporters, and for that, in that sense, we were also isolating new yeast species, especially from the Spataspora clade, that are efficient xylose fermenting yeasts. And we even sequenced the genome of Spataspora arborariae in order to find new transporters. So what we are saying is that here is a picture that shows the classical exhausted transporters from cerevisiae, also the known transporters from Pick's chipiches, but we were looking at other transporters that, as you can see, are quite different from this classical exhausted transporter. And in the genome of Spataspora arborariae and the genome of Passalidarum, we indeed found two genes, which we name it SHUT1 from arborariae or SHUT1 from Passalidarum, which had a high identity with the SHUT1 gene found in chipiches. But curiously, when we express this transporter in the exhausted transporter null strain, they didn't work out, meaning that the cells had the plasmid, but the cells could not grow on glucose, could not grow on xylose. They could only grow on maltose, which is the transported by another sugar permease. So we couldn't understand why it didn't work out. And curiously, when we grew the cells on maltose and used radioactive glucose or radioactive xylose, we could indeed find good transport activities, both for glucose and xylose, with the transporter from chipiches or with the transporter from Passalidarum. So we couldn't, the cells are expressing a transporter, the transporter functions, but it doesn't allow growth on this carbon source. So one of the issues that we were looking at is the possibility that these transporters could be removed from the plasma membrane through ubiquitination. So many transporters have lysine residues either in the end or C-terminal of the protein, and these lysine residues can be ubiquitinated, and that's the signal for removing these transporters from the plasma membrane. So indeed, when we look at the sequence of the genes that we were working, the transporter that we cloned and work out from chipites did not have a consensus ubiquitinated lysine in the sequence, while the new transporters had a highly confident lysine that could be ubiquitinated. So the approach that we took is just to design new primers in order to amplify the gene, but removing the initial part of the gene, including the lysine. So it's what we call a truncated version of the transporter. And as you can see here in the right side, this transporter, the normal transporter, the full-length transporter, does not allow consumption of glucose. The cells will not grow on glucose, will not ferment glucose. But when we transform the cells with the truncated version of the transporter, then the cells will consume glucose, will grow on glucose, and ferment this glucose. The same is true for xylose. The truncated version of the transporter allowed growth on xylose, while the normal transporter did not. And as you can see here, xylose is also very efficiently consumed, producing a lot of ethanol and xylitol, while the transporter with the full length, the gene with the full length transporter did not allow consumption or fermentation of xylose. Another issue that we were looking also is the involvement of alpha ARES genes in this process. These ARES genes, there's over a dozen different ARES genes in cerevisiae. They receive this signal and they recognize the transporters and they recruit the ubiquitinase ligase in order to, this ubiquitinase ligase will add ubiquitin to the lysine residues, and that's the signal for removing the transporter from the plasma membrane. Although the signal sometimes for some transporters is the presence of the glucose or the sugar, while for other transporters it can be the absence of a fermentable sugar. So from the more than a dozen different alpha resistines, we choose two of them, ROG3 and ROD1, which were known as involved in the degradation of some sugar transporters in cerevisiae. So what we did is we delete these arrestins from the genome of the cells. And as you can see, ROG1 did not work out, but when we delete ROG3, then the cells will grow better. In this case, it's on glucose, and they will ferment efficiently this glucose. And that was done with the full-length shoot-1 from Basalidarum or the full-length shoot-1 from Arboriae. So just to end up my presentation, I would like to give the message that for having a successful expression of heterologous sugar transporters in cerevisiae, researchers should look to the presence of lysines capable of ubiquitination. And if the permease has these lysines, they should be mutated or the protein should be truncated in order to allow the stable expression of these transporters in yeast. And obviously, it's also of interest to have or to know which arrestins are involved in this process. Hopefully, this will allow the discovery of new, more transporters, some of them more suited for xylose transport and fermentation. So I would like to thank the PhD students involved in this work, and also Dr. César Fonseca from Portugal, which was also involved in this project, and obviously the funding agencies which allowed us to perform this work.
So thank you very much, the organizers, for this great opportunity to talk at this important conference, the BBS 2021. So my name is André Damasio. I'm from the Institute of Biology at Unicamp, and today I'm going to talk about how N-glycosylation can interfere on enzymes activity and production using a fungal system. So briefly, I want to discuss the importance of enzymes in biorefinery context. So enzymes are central components in this context. For example, converting polysaccharides such as hemicellulose and cellulose in monosaccharides, and these monosaccharides can be used for the production of a wide range of high added value products, including ethanol, for example. And microbial cell factories are the best enzyme producers in nature, especially filamentous fungi, here represented by Trichoderma. So, using these microbial cell factories, enzymes can produce off-site and transported to the ethanol plant, or these enzymes can be produced on-site adjacent to the ethanol plant, or finally, enzymes can be produced integrated to the ethanol plant. So enzymes are produced in the ethanol plant and now using the plant biomass as enzyme inducers instead of glucose like off-site and on-site configuration. But for enzymes production at these integrated configurations, we need very robust microbial cell factories, but it's possible to reduce enzyme costs by around 50% using this configuration. So here it's a good example of a very robust fungal cell factory for enzymes production. It's a Trichoderma rhizii strain from VTT in Finland. So after some decades of genetic engineering, this strain produced around 80 grams of protein per liter, while a laboratorial strain produced 1 or 2 grams of protein per liter. So the point here is how to rational design fungal strains to produce high amounts of enzymes with different biotechnological applications. That's the major question in my laboratory. So we have studied the recombinant enzymes production in fungal cell factories, especially Aspergillus species, and you can see here that it's possible to produce a good amount of recombinant proteins in these strains. In the first two lanes, you can see the reference strains or reference lanes, and we can produce a high amount of xylanase, arabinofuranolidase, cellobiohydrolase, and so on. So these are good systems for enzymes, recombinant enzymes production. But the recombinant enzymes production induce cellular stress in Aspergillus and other fungal cell factories. Here you can see a whole proteome data comparing in the first lane reference strains of Aspergillus nidulans and two recombinant strains producing arabinofuranolidase and cellobiohydrolase, and it's clear that different biological processes are overrepresented in recombinant strains. For example, endoplasmic reticulum stress, energy pathways, oxidative stress, and so on. And by transcriptome data by RNA-seq is also evident that different biological processes are overrepresented in Aspergillus nidulans recombinant strains, and different genes are differentially expressed, and in the last years we are very interested on N-glycosylation pathways and how these post-translational modifications can interfere on recombinant enzymes efficiency and secretion also. So briefly, N-glycosylation is a very conserved post-translational modification and occurs at these protein contacts, asparagine, X, serine, or threonine, and this is the core N-glycan synthesized in Aspergillus, composed by 2 N-acetylglucosamines, 9 mannoses, and 3 glucoses. So we have seen that genes in the N-glycosylation pathways are differentially expressed in Aspergillus nidulans recombinant strains, and also when we induce chemical cellular stress in Aspergillus nidulans using DTT or tunicamycin. So different genes in this pathway are differentially expressed. So that's why we are interested on this pathway on enzyme efficiency and secretion. So some years ago we did the N-glycoproteome of Aspergillus nidulans growing on different carbon sources, and our interest was first in how is the predominant N-glycan in Aspergillus nidulans. So we conclude that N-glycan with 5 mannoses is predominant in this strain, and is rare to find hyperglycosylation forms. And we identified some glycoside hydrolases, actually a lot of glycoside hydrolases that are N-glycosylated in this system, and we selected this target N-glycoprotein. Actually, it is a beta-xylosidase named BXLB, and this target N-glycoprotein has 7 predicted N-glycosylation sites. So here the model of the BXLB, and you can see here the 7 predicted N-glycosylation site, and here you can see the substrate binding pocket of this enzyme. So this is our model enzyme, and based on the wild type protein with again 7 predicted N-glycosylation sites, actually we confirmed all the 7 sites by mass spectrometry, and based on this wild type protein we designed 9 glycomutants, and combining different N-glycosylation sites or maintaining individual sites, and also completely changing the N-glycosylation context by creating new N-glycosylation sites and also completely deleting all the N-glycosylation sites and producing an entire non-glycosylated form of BXLB. So summarizing, here you can see kinetic data for the BXLB wild type and all the glycomutants, and in summary you can see that it's possible to generate enzymes more active only by changing the context of N-glycosylation in these proteins. For 3 mutants, we have enzymes with higher catalytic efficiency than the wild type. So it's possible to generate more efficient enzymes only changing the N-glycosylation system. How about the production of these BXLB glycomutants? By Western blot using a specific antibody for BXLB, we conclude that all the glycomutants are less secreted than the wild type protein. So this is very interesting because yes, we can improve enzymatic efficiency by manipulating N-glycosylation sites, but the enzyme production is reduced in this system, Aspergillus Nidulans, using this model enzyme BXLB. So the conclusions for this first part: genes in the N-glycosylation pathway were differentially expressed under biological and chemical induced cellular stress. Partially glycosylated mutants showed higher catalytic efficiency. All the glycomutants showed lower secretion, and misposition of N-glycosylation sites completely impair the BXLB CC mutant production. So we also asked about the N-glycan synthesis in the fungal system, in Aspergillus Nidulans. So the N-glycan synthesis involves a lot of genes in the endoplasmic reticulum, and this N-glycan is partially synthesized and flipped to the ER lumen, and the core N-glycan finally is transferred to the nascent protein. And this partially folded glycoprotein passed through a very important quality control pathway involving different chaperones, especially connexin, that is a chaperone that binds glycoproteins and assists in the fold of this protein, and this protein can be finally secreted, or misfolded glycoproteins are degraded by the ER-associated degradation system. So we generated a library of Aspergillus Nidulans single mutant strains. Here you can see the reference strain growing on glucose, and here some mutants, some single mutants. Actually, we tried to delete 11 genes in the N-glycan synthesis pathway. In red, you can see probably essential genes for Aspergillus Nidulans, because after 3 rounds of mutations, we didn't recover stable mutants, so probably these genes are essential for Aspergillus Nidulans. And for 2 strains, we got very sick strains, so it was not possible to follow with the next experiments. So in the first round, we obtained 5 stable single mutants for different genes in the N-glycan synthesis pathway. In the second round, we tried to delete also genes encoding for proteins related to the quality control into the ER. So we obtained 2 stable mutants for connexin and glucosidase 2, very important proteins in the ER quality control system. So finally, we transformed the same model glycoprotein we chose before, the BXLB, in the 7 single mutants of Aspergillus Nidulans mutants, and we evaluated the BXLB activity and secretion in each of these single mutant strains. So in this system, the BXLB is under the control of constitutive promoter, So the recombinant protein is induced on minimum medium plus glucose, and this strain anide in black is the reference strain, and this strain does not produce beta-xylosidase on glucose. So here is the Aspergillus Nidulans strain producing the BXLB wild type protein. So in conclusion, when we measured the BXLB specific activity in the reference strain and the single mutant strains, we conclude that BXLB was less secreted in all the single mutants you can see here. When we normalize the specific activity by the fungal biomass, the result is the same. So BXLB is less secreted in all the single mutants of the N-glycosylation pathway. And we specifically measured the secretion of BXLB again by Western blot using a specific antibody. And again, the reference strain did not produce BXLB on glucose, but our recombinant strains, yes, produce in a good quantity. But when we compare the production of BXLB in this strain or in the mutant strains, is interesting because we, at least after 3 experiments, we conclude that the secretion of BXLB is not reduced in the mutants. So the secretion is almost the same, but looking at the specific activity, it's clear that something is not, something wrong is going on in these strains. So, but the deletion of these genes do not affect only the BXLB production, but interfere in all the secretome of Aspergillus Nidulans. Here we measure the total protein in the secretomes in a time course experiment, and we can conclude that the protein secretion was reduced in all the single mutants of Aspergillus Nidulans, showing that the N-glycosylation pathway definitely is important for enzymes production in this system. In conclusion, the second part, genes at the early stage of Aspergillus Nidulans N-glycan synthesis pathway are probably essential for this strain, and the knockout of genes involved in the N-glycan synthesis and processing impairs protein secretion in Aspergillus Nidulans. So thank you very much for the funding agencies. Actually, this work was funded by BioN FAPESP. I also want to thank my very enthusiastic group here at Unicamp and a lot of collaborators around the world and here in Brazil. And thank you again for this great opportunity.